I would like to welcome everyone to Mr. Henderson and Mrs. Gapehart's presentation on meiosis and mitosis. We will begin with mitosis. Interphase, or the living phase of the cell, is the phase in which the cell spends the majority of its time, including preparation for cell division. Interphase is split into three stages: the G1 phase, where the cell grows and goes about normal functions, the S phase, where the cell duplicates its chromosomes, and the G2 phase, where the cell then prepares for mitosis. Prophase is the next stage of mitosis. In this stage, the chromatin condenses into a highly ordered structure called a chromosome. During prophase, the two centrosomes split while increasing their microtubule activity. The centrosomes will later be pushed apart to opposite ends of the cell. In prometaphase, the nuclear envelope breaks into fragments and disappears. Microtubules emerging from the centrosomes reach the chromosomes, moving them towards the center of the cell. Next, we have metaphase. Here the microtubules have finished moving the chromosomes to the metaphase plate in the center of the cell, preparing for duplication of the cell. In anaphase, the microtubules and their accompanying centrosomes then pull their sister chromosomes away from the middle of the cell to opposite ends of the cell. Telophase and cytokinesis are stages in which corresponding sister chromosomes attach at opposite ends of the cell. A new nuclear envelope, using fragments of the parent cellular membrane, forms around each of the separated sister chromosomes. Both sets of chromosomes, now surrounded by new nuclei, unfold back into chromatin, and the cell is pinched off with the metaphase plate. Meiosis, like mitosis, is preceded by the replication of chromosomes. However, this single replication is followed by not one, but two consecutive cell divisions, called meiosis I and meiosis II. These two divisions result in 4 daughter cells instead of 2. In prophase I, the nuclear envelope disappears, and chromatin condenses into chromosomes. The centrosomes also separate to opposite ends of the cell. Metaphase I, like metaphase and mitosis, is where the microtubules, which were released by the centrosomes, have pushed the chromosomes out into the middle of the cell. In anaphase I, the proteins responsible for holding together the sister chromosomes are broken down, and the centrosomes pull their connecting chromosomes to opposite ends of the cell. At the beginning of telophase I, each half of the cell has a complete haploid set of replicated chromosomes. Each chromosome is composed of 2 sister chromatids. One or both chromatids include regions of non-sister chromatid DNA. Cytokinesis, which is the division of the cytoplasm, usually occurs simultaneously with telophase I, forming 2 haploid daughter cells. In animal cells, a cleavage furrow forms at the metaphase plate of the cell. So this concludes our presentation on scorpion-flavored ice cream. Meiosis and mitosis. For more topics like being a fly for a white guy, riding a horse backwards, how to make love in a club, looking talented when you really aren't, hand signs that will get you killed, maintaining manhood while in a sorority house, getting on the most wanted list and staying there, spanking your teammates without leaving the wrong impression, and of course, art history, please visit bioawesomeness.com.